Hello, Health Science 3052 students. This is Professor Van Dyke with your Week 7 video. We have 18 slides for this video. It's not going to be terribly long, and I really want you to watch this video, enjoy it, read the PowerPoint, but I want you to also have time to work on your papers and your presentations. So one of the things I wanted to mention that with your presentation, I've had a lot of questions about how you're going to submit your paper and your presentation to me. You can go ahead and email those to me at william.vandyke@csusb.edu, and I'll go ahead and take a look. A lot of people put them on like a Google Drive and then just send me the link to that because the presentations can be kind of tough to send over email. So keep that in mind. The easiest way to do that is to just go ahead and send it to me with a Google Drive, or if you can just go ahead and send it to me over email. Whatever's easiest for you. I want to make this as simple as possible. I've had some students share their presentation over like a Dropbox video, and I just want you to know that whatever you choose to do is perfectly fine with me. Just make sure you get it to me. Everybody's doing a really great job. I've had a lot of great questions. I started a discussion forum in regard to risk assessment. And again, risk assessment is really something that I want you to all know and understand, and I want you to take it from this class because it really can get you a job. So keep that in mind. And let's go ahead and jump into the pest and pesticide video for week 7. This is pest, pesticides, and vectors, and we're on slide 1. So what is a pest? We're going to talk about pest control, chemical and non-chemical means. We're also going to talk about IPM, which is integrated pest management, environmental impacts of pesticides, pesticide hazards to humans, and alternatives to chemical pest control. So lots of fun stuff. This is my area of expertise. I've worked in vector control for approximately 10 years. I've done every single position from lab, as an unpaid intern all the way up to district manager of Northwest Mosquito. I currently work for Riverside County Environmental Health for vector control. So a lot of fun. We'll go ahead and jump on to slide 2. What is a pest? A pest is defined as an animal, plant, or microbe that adversely affects human interest. And we're talking about pests. We're also talking about vectors. We're talking about a lot of things. But when people think about a pest, they tend not to think about plants. Things like Arundodonax that grows and is called creek cancer, has a very negative effect on waterways like the Santa Ana River Trail. But we're also talking about animals potentially. Animals can very easily qualify as a pest if you think about things like maybe a rabid skunk or a roving gang of raccoons, things like that. But these are going to have an adverse impact on human interest, and certainly microbes can also fit into that category as well. So when we talk about human interest, we can be talking about health, like West Nile virus from mosquitoes. We could talk about clothing that can be eaten up by moths, stored food that may be affected by a microbe like Aspergillus flavus. We could be talking about pests that affect your house, things like termites that can destroy your home. We can talk about all kinds of different things like garden and lawn and trees. Certainly you know about the bark beetle and things like that. Animals, animal products, recreational, agricultural, a lot of agricultural pests, horticultural, with things like spider mites and things like that. All kinds of good stuff here. So pests can include insects and ticks and mites, nematodes, even birds. So you think about insects, we talk about mosquitoes and ticks and black flies and things like that. But we also have lots of other things as well, like birds potentially carrying bird mites, rodents like Rattus rattus, or even the Paramiscus maniculatus that can carry things like hantapulmonary syndrome for people. So this is where hantavirus comes from. Rabbits, weeds, fungi, bacteria, viruses, all kinds of different things that can be labeled as pests. Move on to slide 3. As you can see here with slide 3, the world's deadliest animal by far is the mosquito. And this is certainly a pest or a vector as well. This is I'm going to use those terms kind of interchangeably a little bit, but when I'm talking about vectors, please know that I'm primarily talking about things like mosquitoes. So certainly very dangerous, can cause a lot of health associated issues with not just Certainly not West Nile virus. West Nile virus is very dangerous here in the United States, but you think about malaria that causes an extraordinarily high number of deaths across the planet. So that's a lot of where that 725,000 deaths comes from, is from malaria. On to slide 4. So I'd like to talk about the last time the human population went down. And if you look at slide 4, I'm sure most of you probably know, excuse me, with slide 4, the last time the human population went down was in association with this image. Obviously a very difficult time for humankind. If we jump onto slide 5, we'll see essentially what that dip in the human population was. So the human population has consistently gone on and on and on, and then you see that little small dip there. That is the Black Plague, which is caused by the Oriental rat flea, which came from rodents that were coming off of ships. Most people believe that it was Rattus rattus, the black rat, but it could have also been the Norway rat as well, which is a very good swimmer. And certainly when that came up into the homes with the sailors and things like that, the Oriental rat flea began to spread Yersinia pestis, which is the bacteria that causes plague. So very, very dangerous. Certainly, as you can see here from the numbers, really the last time that human population went down, you see the massive jump in the J curve there at the end, where we're jumping up into the modern age. But certainly a very dangerous time. And you look at this, and this was a vector-borne disease. This was from a pest, and it created extraordinary problems. I try to keep these videos short for you all. If you took my class at a brick and mortar school, you would find that I tend to tell a lot more stories. And I apologize, I have to interject a story here. When they had the Black Plague in France, if you go to France today, you can actually find a large statue of a cat. And the reason for that is that because during the Black Plague in France, they didn't know what was causing it. So what they did was they thought for some reason it was cats, and they went out and started a mass pogrom where they just started killing cats. And as you know, because the Black Plague was spread by rodents, when they killed the cat population, the rodent population exploded, and it made the Black Plague far worse in France. And so you can still find a statue of a cat in Paris, France today. So interesting stuff. On to slide 6. So this is what I like to call the matching game, and I really want you to be able to do this. If you don't know the answers to these questions, make sure you know them, because it's fairly important to me that you have an understanding of what causes what. So the Oriental rat flea, I'll give you a second to look at the list on the right hand side and see if you can tell what it causes, and I'm hoping that you can. So Oriental rat flea causes bubonic plague. The kissing bug causes Chagas disease, which is very common in South America. A very, very dangerous and very deadly disease. It was identified by Carlos Chagas, who later died from Chagas disease. A very horrible thing, and certainly if you have a home that has openings into the house, these kissing bugs can get in. They're called kissing bugs because they crawl onto your face while you sleep. They're attracted to the CO2. They bite you near your mouth, and after they bite you, they defecate in that same place. When you wake up in the morning, you go, oh, why does my face itch? And you're essentially smearing the viral components from the kissing bug into that open wound, and that's going to cause Chagas disease. Really very horrible stuff. And then you have ticks. Ticks are going to cause Lyme disease, most certainly. They're very common. Lyme disease is common in the Midwest and the East Coast. I, of course, skipped over mosquitoes because mosquitoes are the dominant on this list. So mosquitoes are going to cause malaria, Zika virus, chikungunya, yellow fever, and dengue fever, which is all the ones on the bottom. So again, the matching game, you'll probably have a test question or 2 or 3 or 4 on that. So make sure that you know these. Pretty straightforward stuff. On to slide 7. Slide 7 is in regard to pest caused losses, and certainly food crop loss at 30 to 35% from these pests associated with harvest. So a fairly substantial amount, and certainly here in the United States, we lose a lot less because we use a lot of pesticides, but we also use genetically modified crops. So think what you will about GMOs, and a lot of people believe that they're bad, but we are getting a lot more food and a lot less damage to our crops and a lot less um, utilization of pesticides because they are genetically modified to withstand pests and things of that nature. 10% of the pesticides worldwide are used on cotton. We still do grow a lot of cotton, but certainly with synthetic fibers and things like that, we're using much less of that. With the current trends in restricting pesticides, there's about a 25 anticipated percent drop in production, but losses can go as high as 50%. And certainly there are a lot of public health pests and diseases that can be problematic for us here and globally, specifically. So with question, or I should say with slide 7, I definitely want you to know that there are specific public health related pests and disease, and we'll talk a little bit about that here. So we're going to move on to slide 8, public health pests. Now again, these are public health pests. They affect public health, okay? So, I mean, we can talk about things like termites and ants. Certainly, when we think about termites specifically, termites are not a public health pest. They will destroy your house, but they are not a public health pest in the same as they are on slide 8 here, because while it destroys your house, it doesn't vector diseases to humankind. So with slide 8, I want you to focus on that the fact that there is a difference, there is a delineation between pests and public health pests. Okay, a pest may be a termite in that it destroys your house. A moth may be a pest in that it eats your clothes. But on slide 8, you'll notice these are public health pests because they can cause specific things. So pests are going to cause itching and discomfort like lice and fleas and mites and bed bugs, spiders, certainly black widows, house and violin spiders, and they can cause discomfort from their bite, and that can be very problematic for individuals. Certainly fleas can cause things like murine typhus, lice can cause scabies, things like that. And then if you look at the bottom of slide 8, we'll talk about disease vectors specifically. And again, you remember vectors are kind of the same thing as pests. So mosquitoes, flies, body lice, fleas, cone nose bugs, ticks, rodents, bats, birds, etc. So why are these disease vectors? We can look at these kind of one at a time. Mosquitoes can cause what we'd already talked about on the list. Flies can cause onchocerciasis. They can also be a manual mechanical vector transmission processes. So like a black fly can bite you, and it can put a lymphatic filariasis, which is basically a worm, and it can cause very horrible things. So you've seen elephantiasis, where the arm or the leg swells up. That's caused from a fly, actually, a black fly. But you also have body lice that again can cause scabies. Fleas can cause typhus. Ticks, of course, can cause Lyme disease. Rodents can cause all kinds of stuff, including some really horrible things like, of course, the Black Plague. We actually still do have bubonic plague pop up from time to time. Bats can have issues in regard to their fecal contamination, but they can also have rabies as well. And birds can as well. They can pass all kinds of stuff on through their fecal contamination. On to slide 9. So I used to travel quite frequently with the jobs that I've had in the past, and I would find bed bugs more often than you'd think in hotels. And usually the best place to look for them is in the just underneath the edge of the bed. But you can also find them behind the headboard in a hotel room, and you can find them in the pictures behind the headboard. So these are the places that I look for bed bugs. So on the bottom of slide 9, you see an individual here, but how do you avoid doing this if you, obviously most of us don't carry around an insect suit with us. And I've been to hotels and motels in fairly dangerous places like parts of Brazil, parts of Mexico, even parts of the United States. So don't think that just because you're here in the United States that you're safe from bed bugs. I've found bed bugs in some very, very elaborate, very luxurious hotels here in the United States as well. So I tend to look around in my hotel room, and when I find bed bugs, I will request a different room. But sometimes you go to a hotel and they don't have another room for you, and there's no place to stay. There's no other hotels in the area, and so this is going to kind of sound odd, but does anybody know where you would sleep in that situation? In the bathtub. That's right, because bed bugs cannot climb up the side of a bathtub. So if you're making a short stay, a bathtub is actually a good place to put your suitcase and things like that so you don't get hitchhikers that come home with you. And certainly not the most comfortable place to sleep, but when you're desperate, sometimes you got to do what you got to do. So bed bugs can be fairly challenging. They're not a public health vector, but they are certainly a vector of concern because they cause itching and extreme discomfort and cause an infestation in your home. On to slide 10. So what do we have for pest control? We have a lot of different things, and we'll talk about a few of these here. So we have chemical methods, both natural and synthetic, inorganic and organic. So you can have things like pyrethrins, which come from chrysanthemum plants. You can have synthetics like DDT, resmethrin, products like that, and certainly they can come from inorganic and organic sources. We also have non-chemical means like physical means, so I'm going to focus on mosquitoes. A physical and mechanical means could be just dumping out water because, as you know, mosquitoes start their life cycle in water. You can dump that water off or put screens up on your windows. That's a very good physical means. Cultural practices may be avoiding going out during dawn and dusk hours. Source reduction, you may be able to drain a swamp like Washington, D.C. used to be a huge, massive swamp. We drained it. We got rid of a lot of the mosquitoes. Biological and microbial, which is primarily what we use. So we use a bacteria called Bacillus thuringiensis israeliensis that only kills mosquitoes. It's called BTI for short. It works very well. But we also have this sterile male release where, and believe it or not, this actually works in order to kill the mosquitoes that you don't want, which is the females that bite you. You can release sterile males and they will go out and they will compete with the wild population. They will mate with the females, and then the females will have sterile and dead offspring. So this can be very effective as well. We also have things like legislative methods that can be very assisting, but certainly, as you remember, you have the Federal Insecticide, Fungicide, and Rodenticide Act of 1947, which makes us really look at the best means for controlling mosquitoes and other vectors of public health concern. On to slide 11. So these are some means upon which we can chemically control pests. There are inorganic methods that were pre-DDT, so everybody remembers DDT, which is a chlorinated hydrocarbon. Works very, very well as long as you don't develop resistance, and we'll talk about that here at the end. So certainly inorganic and organic methods. And then you have the chlorinated hydrocarbons like DDT, organophosphates like malathion, which we use on medflies still today, and very, very effective. Let's move on to slide 12. Again, we have pyrethrins and pyrethroids. We have insect growth regulators that trick the mosquito larva into thinking that it will stay young forever, and it basically stays in that life stage until it dies. I could give a whole lecture on IGRs. And have a tendency to do so. I can speak fairly regularly about these things. If you have other questions, make sure and let me know. But certainly what you've seen First Exam, and you know that I don't trick you on this stuff. It's pretty straightforward, okay? But we also have rodenticides that we use, like difascinone and warfarin, which cause the rodents to bleed out and die. Do not throw these into your attic assuming that the rodent is going to leave the house and look for water. They don't do that. They feel sick, they bed down, and they'll die inside your attic or inside your drywall. Very dangerous and stinky stuff. So we also have biological methods like predators like mosquito fish, bacteria like BTI, viruses and nematodes and copepods and things like that. And we talked about the other non-chemical means, including like physical and mechanical. So you can put up a window screen that's going to keep them from getting in. You can keep your doors closed. These are all things that you can do. Now, again, this is one of those areas where public health gets affected by socioeconomic status, and certainly when we think about that, it's tough to kind of consider the fact that socioeconomic status is going to play a large effect in regard to how people can protect themselves from vector-borne diseases. Because I know for me, when I was poor, the first thing I did was open up my windows and stop running the AC, especially all night long. That's when the mosquitoes come out. So you can see how that's going to affect an individual or a family of a lower socioeconomic status. Specifically, if I work on a grounds crew, I want to be out early in the morning during dawn and dusk hours when mosquitoes are active, because it's cooler and it's going to be easier for me to do my work. So with IPM, IPM is an integrated pest management philosophy that focuses on combining all these things simultaneously. And really what it means is that when I go out to fight against mosquitoes, I'm going to take my entire toolbox with me, and I'm going to use every tool I have to make sure I can protect people from vectors and vector-borne diseases. That's slide 12. On to slide 13. So what are some of the environmental impacts on pesticides? So we have pest resistance to pesticides. The more we use them, the more the pest will become immune to it. And again, we'll talk about that here at the end. But we also have effects on non-target species, including natural enemies. But you think about, you go out and you kill mosquitoes, or you put neonicotinoids inside your plants that you're getting from hardware stores. Those can affect the bees and potentially can kill bees. So they can be a problem in regard to trying to keep the things safe that we want to keep safe. We can cause pest resurgence and secondary pest outbreaks. So you wipe out all the mosquitoes and then something else breeds up in its place. So that can be a problem. But we also have environmental contamination of the soil, the water, the biosphere, direct applications for crops and spray drift and things like that. So it can have an effect on the environment when we use a lot of pesticides on a regular basis. On to slide 14. Slide 14 is going to show you how DDT essentially generated mosquitoes that were resistant to DDT. And when I was in some third world countries, we certainly did see this, because what you could do is you could take DDT, which they still regularly use. For some of them, they bought all our excess DDT and they still use it. So what you see in population 1, they're treated by DDT, and the only thing that survives is going to be the mosquito that is not susceptible to the toxic power of DDT, and they're going to survive, and they're all going to breed. And then what you're going to end up with is population 2. And when you treat it with DDT, all those mosquitoes survive because you killed all the mosquitoes that were susceptible to DDT, and you end up with the mosquitoes that had no issues with survival. So now all of a sudden they are resistant to DDT. I've seen, I kid you not, I've seen mosquitoes completely covered. They looked like a frosted flake covered with DDT, and it had absolutely no effect on them because they were carrying the markers in their genetic material so that they were not susceptible to the effects of DDT. You keep using the same thing over and over and over again, and this is the problem that you run into: pesticide resistance. On to slide 15. I just love this mosquito, and literally I've seen very similar things to this in the wild. So you can come across this from time to time. That resistance is something that can develop, and certainly with chemical means, it's usually where you see it. On to slide 16, hazards to humans. Certainly effects on farm workers, application reentry intervals, effects of pesticides on these individuals, and pesticide residue in food. So that's why you want to really wash off your crops that you bring in. If you have grapes or strawberries or things like that, you just want to wash them off. But certainly a lot of effects on farm workers. Some of my earlier studies were in regard to workers that worked in the Coachella Valley that were being exposed to pesticides, and they developed higher rates of skin cancer and things like that. So it is absolutely something that does occur, and it's very problematic for those workers. And bullet point 2 talks about re-entry intervals. Here's the deal, okay? When you spray a pesticide over a crop, the workers aren't supposed to go back in for an extended period of time. If they're not working, they're not getting paid. So what do? They go in because they're hurting, and it's a very hard life to be a migrant farm worker. So it's a real challenge, and this is one of those things that has a long-term lasting effect on these individuals and the health of their lives. So it can be very challenging for these individuals to deal with these hazards in association with pesticide use. On to slide 17, we do have some alternatives to using chemicals. There's a lot of different things we can do. We can use natural enemies, we can use pathogens and parasites, we can use attractants to draw them in and trap them, we can use insect growth regulators, we can do the sterile male technique, which we've already talked about, but we can also do crop rotation. So certain crops will draw in things like eye gnats, which cause conjunctivitis, but sanitation as well. So if we keep things clean, then we certainly are in a lot better shape in regard to things like flies. So if you mow your lawn and you leave the grass clippings out, you'll find that you end up with these little green jacketed flies or blue jacketed flies. Those are called bottle flies. And again, they're annoying, but certainly if they land on a dog fecal matter and then they land on your hot dog and you eat it, you can get things associated with like E. coli ingestion that will cause you upset stomach and maybe some diarrhea and vomiting. So there are alternatives to chemicals. So we move on to slide 18, and this is kind of always one of the things I like to talk about here at the end, because on the left you have Avril Lavigne, and on the right you have Justin Bieber. Both of these individuals suffered from a vector-borne disease. They both had Lyme disease, and certainly you can actually find videos of Avril Lavigne talking about this, and it's very sad. So she had Lyme disease, and her doctors kept misdiagnosing her and saying, yeah, you're just lazy, get out of bed, go write a song, come on, don't be so lazy, you're rich, come on, get out of bed. And it turned out she actually had Lyme disease that was misdiagnosed time and time again. So what I tell you is this: there is a socioeconomic factor associated with identification of vector-borne diseases. But Avril Lavigne, trust me, she has better doctors than we do. I have Kaiser, and Kaiser's great. But Avril Lavigne's not going to Kaiser Permanente, okay? She has a team of doctors, and certainly they had an interest in making sure that she was healthy. They could not identify that she had a vector-borne disease. She had Lyme disease. She suffered with it. It was absolutely horrifying. And if you listen to her story, it's really quite compelling. You can actually find it. I believe it's still on YouTube. But again, vector-borne diseases can be very dangerous, and they can be very problematic. With the right amount of vectors, they could take over the world and change our lives irrevocably forever. And certainly we saw what happened with Zika virus and how it affected the Olympic Games. That was a vector-borne disease. So we have a tendency to overlook these until they become a problem and blow up in our face, like certainly with the Black Plague. But again, with vector districts across the state of California and across the country, they do the very best they can. So we do have people protecting us from these things. I like to call vector disease surveillance and districts, they really are kind of one of the unsung heroes of public health, and certainly they do a wonderful job at what they do. So this is your week 7 video. Please, definitely get going on your paper and your presentation. This video is designed to be fairly light because I wanted you to be able to focus on your paper and your presentation. Make sure you know what you're writing about and get started on it because it's going to come up pretty fast. And certainly if you look at the syllabus, you'll see that exam 2 is coming up fairly quickly too. So make sure you're ready for that. And with that, again, your paper and your presentation, you'll be able to email those to me at william.vandyke@csusb.edu. That's william.vandyke@csusb.edu. Keep your questions coming. Ask me about risk assessment. Send me your examples, and I'm always happy to talk about those with you. And I hope with that you have a wonderful week. Thank you so much, and look forward to talking to you soon.